students, and today we are going to discuss the questions we can expect from the topic the photosynthesis in higher plants. Okay, see the first question. So NADPH is generated through, and see the choices: photosystem 2, anaerobic respiration, glycolysis, and photosystem 1. Okay, so the question is about the NADPH formation in the photosynthetic process. As we have seen in the introduction of the photosynthesis, there are two major systems involved in the system of the light reaction system, photosystem 1 and photosystem 2. Okay, so they have their own the pigments, and they are going to accept the light energy in the form of photons, and they are going to excite, and the excited energy they are going to activate the molecules. Okay, and they are going to produce the release of electrons, and those release of electrons or released electrons entered into the cycle of molecules or complexes, and they are going to produce the energy rich carriers of molecules like ATP and NADPH. Okay, so this is the common thing you have to remember about the photosynthesis. There are two major reactions as we have discussed: light reaction and dark reaction. In case of light reaction, they are going to produce the energy rich molecules of ATP and NADPH. In case of dark reaction, and they are going to use the particular molecules ATP and NADPH will be used in the dark reaction to produce the carbohydrate in the dark condition by using the enzymes. Okay, so this is what you have to remember. And the question over here is not talking about the ATP and NADPH, it is talking about only the NADPH. Okay, so in the light reaction we have seen two different kind of reactions. Okay, so whenever you are going to produce the high energy molecule, we are calling that reaction as the energy producing reactions. And over here we have seen two different cycles: cyclic phosphorylation and non-cyclic phosphorylation. Okay, so over here, what do you mean by non-cyclic phosphorylation? Over here the electrons will not be backed, or will not be in a cycle to the particular reaction, and it will be delivered to the production of NADPH. Okay, so in case of cyclic phosphorylation, there will be no production of the particular NADPH, because over here there will be no involvement of enzyme called NADP dehydrogenase. Okay, so in the absence of the particular enzyme and in the absence of some other system, over here cyclic phosphorylation going to produce only the ATP molecule and not NADPH molecule. Okay, and this is the second point you have to remember. And the question over here, it is talking about NADPH generated through. Okay, so over here you have some keywords: photosystem 1, photosystem 2, and anaerobic respiration and glycolysis. Okay, and you can easily eliminate the glycolysis and anaerobic respiration. You have to choose from only the PS2 or PS1. As I told you, there are two major reactions over here: cyclic and non-cyclic phosphorylation. So what do you mean by non-cyclic phosphorylation? Over here, the involvement of PS2 and PS1. This is what you have to remember in case of non-cyclic phosphorylation. So what happens over here, as I told you, starts from the light. The photons will be absorbed by the pigments of the photosystem 2. Okay, and it is going to excite, electrons going to release from here. Okay, and the electrons will be accepted by the electron acceptor, and then it will be transferred to the photosystem 1. Over here also there is a reaction center. It is going to absorb the particular electrons released by the particular PS2, and in between the PS1 and PS2 there is a electron transport chain, and that is going to produce the ATP molecules. That is what you have to remember over here. Okay, and the point you have to remember over here, in the PS1 again it is going to accept by the electron acceptor over here, and these electrons, okay, the electrons accepted by the particular PS1 system will be delivered to, or it will be transferred to the final acceptor molecule NADP plus over here, and it is going to produce NADPH molecule over here. Okay, so the point you have to remember over here, the answer for you over here, the PS1. Why? As I told you, here only it is going to produce the particular NADPH molecule. Suppose you have a choice over here like PS1, PS2 and for example instead of glycolysis you have non-cyclic phosphorylation. You have to choose non-cyclic phosphorylation because the NADPH is the product of non-cyclic phosphorylation. In case of cyclic phosphorylation, there will be no production of NADPH, only the production of ATP. This is what the major differences you have to remember in between the particular production of ATP and NADPH. Okay. And now see the next question. Number of Calvin cycles required to generate one molecule of hexose. Okay. So it is talking about a production of one sugar molecule. Over here hexose. Hexose means 6 carbon sugar. For example, glucose is a hexose. Okay. So production of one molecule of hexose requires how many number of Calvin cycle? Okay. And we have seen Calvin cycle. Calvin cycle is nothing but C3 cycle. It will happen only in the C3 plants. Okay. So what will happen over here? What do you mean by Calvin cycle? It is a kind of the carbon dioxide fixation. Okay. Over here the carbon dioxide from the atmosphere it is going to be fixed as a sugar. Okay. So over here it's a continuous cycle of oxidation and reduction reactions. Oxidation and reduction reactions. Okay. So what happens over here? Oxidation and reduction. As I told you, so there are two major molecules involved over here. As I told you, carbon dioxide and water. Okay. So here you have to remember one thing. The water is being oxidized and the carbon dioxide is being reduced. This is what you have to remember. Water is being oxidized. Carbon dioxide is being reduced. Okay. So what do you mean by reduction over here? The addition of hydrogen atoms over here is called reduction. The removal of hydrogen atoms from the water molecule is called oxidation. Okay. So over here oxidized means the water will be oxidized to release oxygen. Okay. So this is what you have to remember. Removal of hydrogen and leads to the release of oxygen from the water molecule. So over here the carbon dioxide, the addition of hydrogen atoms over here, it will synthesizes the carbohydrate from the carbon dioxide. This is what you have to remember. Whenever you are thinking about a carbon dioxide fixation, you have to remember one thing. This is a reaction. This is a continuous chain reaction of oxidation and reduction. So over here there are two major molecules involved, water molecule and carbon dioxide. Water is being oxidized, carbon dioxide is being reduced to the carbohydrate or sugar molecule. Okay, the question over here for you, what happens during the C3 plants or C3 cycle or the Calvin cycle? It has been discovered by Melvin Calvin. Okay, so as I told you over here, a carbon dioxide molecule is being fixed in a particular Calvin cycle. A carbon dioxide molecule enters into the C3 cycle or Calvin cycle. Okay. So the carbon dioxide molecule it is being accepted by the ribulose bisphosphate. Okay, and it will enters into the particular cycle, and at one stage it will produce it will fix one carbon into a organic form. Okay, just remember one thing. The carbon dioxide as a gas is an inorganic form. Over here, when it is combined with a particular hydrogen atom, it is going to be fixed as a organic molecule as a sugar. But the point you have to remember over here, in each carbon cycle or each Calvin cycle, one carbon will be fixed in the form of organic form. Okay, this is what you have to remember. In each Calvin cycle, only one carbon will be fixed in the particular cycle. Okay, so the point you have to remember over here, as I told you, ribulose bisphosphate over here is a 5 carbon sugar. Okay, it will accept the particular carbon dioxide. Carbon dioxide has 1 carbon. Okay, 5 carbon plus 1 carbon, it will synthesizes 6 carbon intermediate. Okay, this is a temporary molecule. This 6 carbon molecule immediately will form 2-3 carbon molecules. Okay, and it will enters into the formation of the organic compound. This is what you have to remember. The point you have to remember, where as I told you, In each cycle it will fix just one carbon at a time. Okay. The question over here, number of carbon cycles to generate one molecule of hexose. As I told you, hexose is a 6 carbon molecule. So you need 6 cycles to fix the particular hexose molecule or to form a particular glucose. One glucose, that is 6 carbon, you need 6 cycles of Calvin to perform. Okay, so you can easily eliminate 4, 9 and 8. Okay, this is how you have to answer the particular type of question from the Calvin cycle. Okay, so whenever you are thinking about a carbon dioxide fixation in case of C3 plants, you have to remember one thing: each carbon dioxide will generate only one carbon fixed in the particular organic form. And see the next question. In C4 plants, carbon dioxide combines with, and see the choices: phosphoenol pyruvate, phosphoglyceric acid, phosphoglyceraldehyde and ribulose bisphosphate. So we have discussed the topic called carbon dioxide fixation. Okay, as I told you, the first discovered cycle in the carbon dioxide fixation is Calvin cycle. Who has discovered the particular Calvin cycle? As I told you, Melvin Calvin. Okay, and the point you have to remember over here, the Calvin cycle, we are calling the plants which perform the Calvin cycle are C3 plants. So why we are calling these as C3 plants? As I told you over here, the first forming product from the particular carbon dioxide fixation. So what happens over here in both the C3 and C4 plants? What happens over here? The carbon dioxide fixation gonna happen. Okay, so they are gonna fix the atmospheric, the gaseous form of carbon dioxide into the organic form of sugar. Okay, this is what gonna happen in the C3 and C4 cycles or C3 and C4 plants. But the question over here, what is the difference between the C3 and C4? Okay, so in case of the carbon dioxide fixation, the carbon dioxide enters into the cycle and it will be fixed as a organic sugar molecule. Okay, so this is what you have to remember. The question over here, carbon dioxide combines with. Okay, so you have to remember the difference between the C3 and C4. Why we are calling the particular cycle as C3 and why we are calling that as a C4. Okay, so C3 means it refers to the first formed molecule from the particular cycle is 3 carbon molecule. This is what you have to remember. Okay, so in case of C3 cycle, the first formed molecule will be phosphoglyceric acid. Okay, so it will be a 3 carbon molecule. In case of C4, the first formed molecule will be 4 carbon molecule, that is oxaloacetic acid. In case of C4 plants, the first forming molecule over here is oxaloacetic acid. It is a 4 carbon molecule. Okay, so the question over here, what will be the acceptor molecule for the particular carbon dioxide in both C3 and C4? Okay, so you have to remember one thing while we are discussing the previous question. As I told you, in case of C3 plants, the carbon dioxide will be accepted by ribulose bisphosphate. Okay, it is a 5 carbon molecule. It is going to accept the carbon dioxide. So it will produce a 6 carbon intermediate. From this it is going to produce 3 carbon molecules. Okay, in case of C4 plants, first acceptor molecule for the particular carbon dioxide will be phosphoenol pyruvate. Okay, there will be no involvement of ribulose bisphosphate as a first acceptor, primary acceptor for the particular carbon dioxide molecule. Okay, so see the question over here, C4 plants carbon dioxide combines with, you can easily eliminate the ribulose bisphosphate. As I told you, ribulose bisphosphate is a primary acceptor of carbon dioxide in case of C3 plants. Okay, so your question is about C4 plants, so you can easily eliminate the ribulose bisphosphate. Thus remember one point, ribulose bisphosphate is also one of the major enzyme involved in C4 plants also, but these are not the particular primary acceptor. Okay, they are involved in the particular C4 plants but in a second phase of the particular cycle. And phosphoglyceraldehyde is not a correct answer. You must choose phosphoenol pyruvate and phosphoglyceric acid. As I told you, phosphoglyceric acid is a product formed from the C3 cycle, and the answer over here, phosphoenol pyruvate. Okay, this is how you have to choose the answer for this kind of question. And see the next question. In C4 plants, carbon dioxide fixation is done by. Okay, this is also related to the previous question. This question is talking about the location at which the particular carbon dioxide fixation gonna happen. Okay, and sclerenchyma cells, chlorenchyma cells, mesophils and guard cells. Okay, so we all know we are talking about the photosynthesis, and it should be the cells which are related to the particular photosynthetic system. Okay, the sclerenchyma, chlorenchyma, they are all related to the storage function of the particular plant, so you can easily eliminate the particular type of cells, and the answer must be in between mesophils and guard cells. Okay, so what are guard cells? As we all know, in case of the leaves, okay, they have some apertures or opening, okay, stomata opening, and every stomata will be surrounded by two cells, and those cells are called guard cells. Okay, so they are involved in the opening and closing of the particular stomatal apertures. But the question is about the carbon dioxide fixation. Okay, so the carbon dioxide fixation, the cells involved in the carbon dioxide fixation are the mesophil cells, not the guard cells. Okay, so you have to choose the answer mesophil cells. And what and all the process involved in the mesophil cells, we are going to discuss those later while we are discussing some other questions in the same topic. Now see the next question. The reaction center during the photosynthesis. Okay, so it is talking about the word reaction center. Carotene pigments, phytochrome pigments, P700 and cytochromes. Okay, and we will recall the particular reaction center. As I told you, the entire photosynthesis is going to start with the absorption of particular photons from the sunlight, that is light energy. Okay, so the photons will be observed over here by means of pigments involved in the particular photosystem 1 and 2, PS 1 and 2. Okay, so the point you have to remember over here, as I told you, as we have seen in the introduction classes, they are going to observe the particular light wavelength in between the 600 and 700. Okay, that is in the visible range. In the visible range, normally they are going to observe red and blue light regions. Okay, and this is what you have to remember. 600 to 700 nanometer range they are going to observe, and for each system, for PS1 and PS2, they have their specific reaction center. So reaction center, which means over here they have some specific or major pigment they are going to observe, or they are going to involved in the absorption of particular light energy. Okay, as I told you, it's made up of so many pigments. Chlorophyll A, B, xanthophylls, okay, there are so many pigments over here. But the point you have to remember over here, all the energy, all the photons is being absorbed by the particular entire pigment system will be transferred, or it will be relocated to the particular reaction center. There are two reaction centers are there, P700 and P680 for each PS1 and PS2. Okay, so this P700 or P660, they are going to act as a reaction center, and they are going to absorb the particular energy, and they are going to excite the particular molecules, and with this action only they are going to release the particular electrons for the further cycle in the photosynthesis. Okay, so the point you have to remember over here, the reaction center, it is always talking about the pigment system, but over here they have given you choices as pigments. Phytochrome is a pigment, carotene is a pigment, but you have to choose. Whenever the choice P700 or P680 is available, you have to eliminate the other pigments. Okay, so the reaction center, whenever you are talking about the reaction center, you have to think about P700 or the P680. Okay, so this is the answer for the particular type of question. Reaction center over here is P700. Okay, you may get the question specificity. What kind of the P center or reaction center in photosystem 1 or what kind of reaction center in photosystem 2. Okay. So in those type of question you have to answer whether it is P680 or P700. Okay. And see the next question. So over here a photosynthesizing plant is gonna release O18 more than a normal oxygen. Okay, so this is your question and this is your statement. The question over here, the plant must have been supplied with. Okay, so it is talking about a process of the photosynthesis. Okay, so over here the particular plant, the particular photosynthesizing plant, it is going to release oxygen in a form of O18. Okay, so what is the normal form of oxygen? O16. Okay, so over here it is going to release oxygen isotope O18. Okay, the question over here, the plant must be supplied with what kind of molecule in a list over here and which molecule or which substitution going to produce the particular O18 as the product or evolution from the particular photosynthesis reaction. And see the choices O3. O3 is nothing but a ozone, and a second choice water with O18 isotope, carbon dioxide with O18 and sugar with O18. Okay. And you have to remember one major point in case of photosynthesis. What is photosynthesis? Here we are gonna utilize carbon dioxide and water and they are gonna produce sugar. This is what you have to remember. Whenever you are thinking about a photosynthesis, carbon dioxide will be utilized, water will be utilized to produce the particular sugar molecule. Okay. So reactants are carbon dioxide and water, products are sugar. Okay, so supplied with is talking about the reactants, so you can easily eliminate the sugar is being substituted with a particular oxygen because sugar is the product going to produce from the particular photosynthesis, you can easily eliminate. And ozone will not be the correct answer because there will be no the isotopic differences mentioned over here, so you can easily eliminate the ozone. The question over here is in between the water and carbon dioxide. What is the statement given for you? The oxygen is being released as O18. Okay. So the question must be like this. What is the source of the particular released oxygen from the particular photosynthesis? We all know during the photosynthesis the plant gonna release the oxygen or the oxygen will be evolved from the particular photosynthesis. The question over here, what is the source of the particular oxygen, whether it is a carbon dioxide or it is a water molecule? This is the straight question from the particular topic. Okay, so both the carbon dioxide and water will contain the oxygen atom. Okay, so the question over here, what is the actual source of the particular oxygen for the particular photosynthesis reaction? Okay, so over here you have to remember one thing. Among these two choices, water with O18 or carbon dioxide with O18. Okay, so the key point you have to remember in the photosynthesis, the oxygen evolution from the photosynthesis is from the splitting of water. Okay, so as I told you, the light, they are going to release the photons, the excitation of photons, they are going to release the excitation of molecules, they are going to release the electrons. Okay, the water will be split into molecule, the atoms. Okay, H plus protons plus electrons plus oxygen. Okay, these are all the products from the water splitting. Okay, splitting of water leads to protons plus electrons plus oxygen. As I told you, the protons and the electrons will be utilized for the synthesis of ATP and NADPH in the light dependent reaction. The oxygen will be released from the particular reaction, water splitting. Okay, so the answer over here is the water is being labeled with the particular oxygen isotope. Okay, so the carbon dioxide, what may be the use of carbon dioxide? As I told you, the carbon dioxide is going to be fixed or the carbon dioxide is going to be reduced. That means you're going to add hydrogen atoms so that you're going to synthesize carbohydrate from the carbon dioxide. Okay, so the oxygen atoms in the carbon dioxide gonna utilize for the formation of sugar molecule whenever you are observing the product. Okay, suppose you are going to use the carbon dioxide is being labeled with oxygen 18, you will see the particular radio labeled oxygen in the sugar molecule, not in the evolution of the free oxygen from the particular reaction. Okay, so the answer over here is water with O18. Whenever you are using a water with O18, you will see the particular oxygen in a released gas form from the particular photosynthesis. Okay, and see the next question over here. The PS2, okay, photosystem 2 occurs in. Okay, so this is talking about the site or location of the particular photosynthesis and photosystem 2. So as I told you, there are two types of reaction in the photosynthesis: light dependent and dark reactions. Okay, so both the reaction going to happen at a organelle called chloroplast. We all know that chloroplast is a photosynthetic organelle of any system. So over here the chloroplast going to involved in the photosynthesis, and it is a organelle, that means a double membrane bound structure. So within a particular membrane bound structure there is a semi-liquid matrix called stroma. Within a stroma there are structures called lamella, and within a lamella there are the coin-like arrangement called thylakoid, and the thylakoid going to form arrangement of grana. Okay, and you have to remember one thing. I told you light and dark reaction. All the dark reaction or the enzymes specific for the dark reactions are present in only in a stroma or liquid matrix of the particular chloroplast. Okay, all the light dependent reactions are present only in the membranes of the particular center organelles. Okay, so this is what you have to remember. Only the membranes are going to participate in the light dependent reaction. All the dark reactions going to participate or they are going to present in a particular stroma or semi-liquid matrix of the particular chloroplast. What will happen during the light reaction? As I told you, absorption of light, splitting of water. Okay, and they are going to produce the ATP and NADPH during the light reaction. What will going to happen in a dark reaction? They are going to utilize the ATP and NADPH to fix the carbon dioxide as a organic sugar molecule. Okay, and see the choices over here: stroma, cytochrome, grana and mitochondrial surface. You can easily eliminate the mitochondrial surface. This is completely different organelle from the photosynthesis. And see the choices, remaining choices: stroma, cytochrome and grana. Cytochrome is a kind of molecule or complex involved in the electron transport chain. Okay, so you can easily eliminate this. The answer choice must be in between stroma and grana. I told you stroma is always involved in a dark reaction. There will be no relation between the PS2 and dark reaction. So you can easily eliminate the stroma. The answer choice must be the grana. Grana is nothing but the modification or the arrangement of the thylakoid one above the other. They are going to form the grana, and in the grana, the inside, the inner side of the membrane possesses the particular PS2 system. Okay, so the photosynthesis system, the photosystem 2 occurs in the grana or the membranes of the thylakoid. Inner side of the thylakoid membrane will have the particular PS2. And see the last question over here: the enzyme that catalyzes the carbon dioxide fixation in C4 plants. This is the clue for you. And choices: ribulose bisphosphate carboxylase, ribulose diphosphate, phosphoenol pyruvate carboxylase and carboxy dismutase. Okay, and this is talking about an enzyme, so you can easily eliminate this. This is not an enzyme. So there are three remaining choices: ribulose bisphosphate carboxylase, phosphoenol pyruvate carboxylase and carboxy dismutase. So while we are discussing the questions in a previous section, as I told you, there are two types of plants, C3 and C4. In case of C3 plants, okay, the receptor molecule or acceptor molecule for the carbon dioxide is ribulose bisphosphate. Over here it is phosphoenol pyruvate in case of C4 plants. So this is a molecule going to accept the carbon dioxide. The enzyme which catalyzes the reaction over here is ribulose bisphosphate carboxylase. So other name for this is Rubisco. Okay, so why we are calling this as Rubisco? Ribulose bisphosphate carboxylase and oxygenase. That is why we are calling this as Rubisco. So over here, as I told you, the primary acceptor is not a ribulose bisphosphate. Over here is phosphoenol pyruvate. So the phosphoenol pyruvate, this reaction, the carbon dioxide fixation by the phosphoenol pyruvate will be catalyzed by the enzyme PEPCO. PEPCO refers to phosphoenol pyruvate carboxylase and oxygenase. Okay. So the question over here is the enzyme that catalyzes the carbon dioxide fixation in C4 plants. In case of C4 plants, you have to always remember one thing. The primary carbon dioxide acceptor over here is phosphoenol pyruvate. So answer must be, you have to easily eliminate the ribulose bisphosphate carboxylase, carboxy dismutase you can easily eliminate. The phosphoenol pyruvate carboxylase or other term, name for this is PEPCO. Okay, so the PEPCO or the phosphoenol pyruvate carboxylate is the answer for the particular type of question. So in the next class we are going to continue the discussion of the same topic, the photosynthesis in plants. Dear students, and today we are going to continue the same section, the photosynthesis in higher plants. And see the first question today. The chromatophores takes part in, and there are choices. Okay, so this question is talking about the chromatophore or the role of chromatophore in the particular plant system. So movement, respiration, photosynthesis and growth. Okay, so this is talking about a chromatophores. Okay, so chromo or chromato, that means color. Okay, so colored phores refers to something called pigments. Chromatophores means the structures they are going to possess the pigments. Okay, structure with pigments. Okay, so that means colored pigments. Okay, so the chromatophores which contains the colored pigments. So what may be the role of pigments? They are going to observe the colored pigments, means they are going to observe. So as we have seen in the introduction classes, they are going to observe the pigments. They are going to observe the light photons in a particular nanometer. Okay, so for each and every pigment they have some specificity to observe the light. Okay, so they are going to involve in that. The colored pigments always going to involved in the absorption of light, so that they are involved in the photosynthesis. Okay, so they don't have any role in respiration and movement and growth. They are involved in the particular synthesis, photosynthesis. And see the next question. C4 plants more efficient in photosynthesis than C3 plants. Okay, so that statement we all know. C4 plants are more efficient than the C3 plants in the photosynthesis. The question is why? What is the reason behind that? Okay, and there are choices. Higher leaf area, presence of large number of chloroplast, presence of thin cuticle and a lower rate of photorespiration. Okay, and see the choices. Higher leaf area, this may be the correct choice when you don't have any other choices, but over here this is not the better choice, so you can easily eliminate this. Okay, so only the higher leaf area cannot be converted into the higher synthesis of or higher rate of the particular photosynthesis. And presence of large number of chloroplasts and presence of thin cuticle. Okay, so you can easily eliminate the presence of thin cuticle, and the choice in between the two, lower rate of photorespiration and the presence of large number of chloroplasts. We are talking about the photosynthesis, and of course the presence of large number of chloroplasts will increases the rate of photosynthesis. Okay, but the point you have to remember over here, when you don't have the choice like this, lower rate of photorespiration, you have to choose the choice. Presence of large number of chloroplasts will increases the rate of photosynthesis. But when you have a choice like this, lower rate of photorespiration, you can easily eliminate this one. Okay, so I'll explain why we have to choose this lower rate of photorespiration. Because what is photorespiration? As I told you in the previous classes, photorespiration, okay, is going to happen only in the C3 plants. So we are talking about the enzyme ribulose bisphosphate, okay, rubisco. So SCO refers to carboxylase and oxygenase. Okay, so this is a activity of the particular enzyme, rubisco enzyme. Okay, so what is the action of particular enzyme? They are going to involved in the fixation of carbon dioxide. So what is the acceptor over here in case of C3 plants? Ribulose bisphosphate. Okay, and as I told you, this enzyme has a specificity for, or they have the affinity for both the molecules of carbon dioxide and oxygen. Okay, but the affinity for carbon dioxide is more than the particular oxygen molecule. So what happens when the oxygen concentration is more in the environment? The oxygen go and binds to the particular enzyme. So what will happen over here? Here the carboxylase activity get reduced and the oxygenase activity of the particular enzyme got increased. Okay, so what will happen? So over here, when the oxygen gonna integrated into the particular enzyme with the help of the oxygenase activity, the oxygen assimilation or oxygenation gonna occur in the particular enzyme. So what will happen? The assimilation or the fixation of carbon dioxide gonna get reduced. So what will happen over here? The oxygenation of the particular oxygen leads to the release of, okay, there will be no release of ATP, just one ATP molecule will get released over here and formation of glycolic acid. Glycolic acid gonna form and this reaction is called photorespiration. Okay, so where it gonna occur? As I told you, it is gonna happen only in the C3 plants because primary acceptor over here is ribulose bisphosphate and the primary enzyme over here is ribulose bisphosphate, carboxylase and oxygenase, rubisco. Okay, so because of this activity of the particular rubisco, because of this photorespiration, okay, so over here in case of C3 plants, their overall photosynthesis rate will be compared to C4 plants, they will be very lower than the particular C4 plants. Okay, so the choice over here, as I told you, the lower rate of photorespiration in case of C4 plants. Okay, why? As I told you, in case of C4 plants, the primary acceptor for the carbon dioxide is phosphoenol pyruvate. So the enzyme involved over here is phosphoenol pyruvate, carboxygenase and oxygenase, pepco enzyme. So for this enzyme, okay, so they will be more efficient in lower concentration of carbon dioxide. So what will happen? As I told you, even though the concentration of the particular molecule is lower in the environment, it will accept only the carbon dioxide more efficient than the oxygen. So what will happen? As I told you, very negligible amount of photorespiration in case of C4 plants. So that will increases the assimilation or fixation of carbon dioxide more efficiently than the C3 plants. So the photosynthesis rate over here in case of C4 plants are higher than the C3 plants. This is how you have to understand the particular area and you have to answer this lower rate of photorespiration. As I told you, when you don't have the particular choice related with the photorespiration, you have to automatically choose the large number of chloroplast because in case of C4 plants, they have large number of chloroplast also in the particular mesophyll cells. We are going to discuss the thing later, but the point you have to remember over here, in case of C4 plants, they have lower rate of or very negligible amount of photorespiration compared to the C3 plants. So their photosynthesis rate is higher than the C3 plants. And see the next question over here. In C4 plants, okay, the malic acid synthesis happens at, or the malic acid synthesis during the carbon dioxide fixation occurs in, okay, so this is talking about the location of the particular malic acid synthesis. As I told you in the previous question, as I told you, there are two types of cells involved in the particular carbon dioxide fixation. Okay, so over here in case of C4 plants, okay, normally C4 plants occurred in, you can observe the particular plants in dry areas, dry tropical areas, okay, and they have large biomass productivity, okay, like sugarcane and maize, okay, they have large biomass productivity. So what will happen over here, they have to synthesize more product by means of the particular photosynthesis process. Okay, so compared to C3 plants, they have more productivity. This is what you have to understand. Okay, so they have to synthesize more product by means of photosynthesis. Okay, so they have some anatomy as we have discussed in a previous class, Kranz anatomy. So what is Kranz anatomy? Around the particular vascular bundle. So vascular bundle we all know, xylem and phloem are vascular bundles. So around the vascular bundles, there are dense cells of the packing. Okay, so these dense cells to packing are called Kranz anatomy. Okay, so over here in case of Kranz anatomy, large number of, as I told you in a previous question, large number of chloroplasts are present in a particular mesophyll cells. Okay, this is what you have to remember in case of Kranz anatomy. Around the particular vascular bundle, the cells are packed in a tight condition. Okay, intercellular space will be very less over here in the Kranz anatomy. Okay, so mesophyll cells are tightly packed. Okay, so behind the mesophyll cells there is a vascular bundle. Sorry, there is a bundle sheath cells. Okay, and both the type of cells gonna involved in the carbon dioxide fixation. So as I told you, carbon dioxide from the atmosphere, it is being primarily accepted by phosphoenol pyruvate. Okay, and with the enzyme, with the help of enzyme, phosphoenol pyruvate co, that is PEPCO, and it will be converted into 4 carbon containing compound, that is oxaloacetic acid. Immediately it will be converted into some other 4 carbon containing compound, that is malic acid. Okay, and this malic acid will be transported to the bundle sheath cells, and over here the further carbon dioxide fixation gonna continue and the cycle will be come back to the particular phosphoenol pyruvate. The point you have to remember over here, the synthesis of malic acid, the location, that is what your question. In leaves, the C4 plants, malic acid synthesis occurs in. Okay, so this question is talking about the location of synthesis of malic acid. You are not supposed to get confused with the term malic acid, because both the malic acid and the oxaloacetic acid both are formed in the mesophyll cells of the particular C4 plants. Okay, so only after the formation of the malic acid, the product, the malic acid will be transported to the bundle sheath cells. Okay, so over here you can easily eliminate the bundle sheath cells. As I told you, guard cells you can easily eliminate, epidermal cells they nowhere related to the particular question. The answer over here is mesophyll cells. As I told you, in the mesophyll cells, the carbon dioxide fixation, formation of oxaloacetic acid, the conversion into malic acid, all the things gonna happen in the mesophyll cells of the particular C4 plants. And see the last question now. The cyclic phosphorylation results in the formation of. Okay, so this is talking about the phosphorylation. As we have discussed in the introduction classes, there are 2 types of phosphorylation, cyclic and non-cyclic. Okay, and over here what may be the product of these cycles? Okay, this is what the question. In cyclic phosphorylation, what is the result or what are the products from the particular cyclic phosphorylation? So as we have discussed in the introduction classes, as I told you, there are 2 types of photosystems, PS2 and PS1. Photosystem 1 and photosystem 2. So as I told you, all starts from the light source and they are going to observe the light with the photon acceptor, the pigments. Okay, and the pigments going to excite the electrons and the electrons going to accepted by electron acceptor, and it will be entered into the electron transport chain. Okay, and this system is called photosystem 2. And from the electron transport chain, the electrons transported to the pigments of photosystem 1, again electrons released, again electron acceptor, then it will be transported to NADP and it will form the NADPH. Okay, and this is what going to happen in a non-cyclic. So the point you have to remember over here, in case of non-cyclic phosphorylation, both the PS1 and PS2 gonna involve. This is what the first point you have to remember. In case of non-cyclic phosphorylation, both the PS1 and PS2 involved in the process. And the second thing you have to remember, over here the electrons are in a non-cyclic manner because over here they are not gonna come back to the particular cycle. They are being transported to the final molecule NADPH. Okay, so from the particular light source and from the molecules of the particular photosystem 2, and the electrons transported to the electron transport chain and to the photosystem 1 and finally to the NADP, and they will synthesize the particular NADPH molecule. Okay, so the point you have to remember over here, during the electron transport chain they will also synthesize ATP molecule. Okay, and this process is for non-cyclic. But in case of cyclic, okay, the cyclic gonna occur, cyclic phosphorylation gonna happen only in the PS1 system. Okay, so you can easily eliminate the particular PS2. Okay, so over here directly from the light source, the photons will be absorbed by the particular PS1 system. So the point you have to remember over here, in case of PS1, in case of cyclic phosphorylation, the enzyme NADP dehydrogenase, okay, is absent. This is what you have to remember for the cyclic phosphorylation. The particular enzyme is absent, that means you cannot synthesize the particular molecule NADPH. Okay, so what will happen? So after the absorption of light, the excitation, electron formation, the electrons will be accepted by electron acceptor and it will be transferred to the electron transport chain, and again the electrons will be come back to the particular cycle. Okay, so what will happen? Over here in case of the electron transport chain, only the ATP molecules will be synthesized and not the NADPH molecule. Why? As I told you, over here the NADP dehydrogenase enzyme is absent, so they cannot convert the particular electrons and protons into the particular NADPH molecule. Okay, so the point you have to remember over here, in case of cyclic, okay, the molecule produced over here is ATP. And the second major point you have to remember over here, the photolysis of water. Okay, as we have discussed in previous classes, photolysis of water leads to evolution of oxygen plus the release of protons and some formation of electrons. Okay, and these things gonna happen only in the photosystem 2. That means in case of cyclic phosphorylation, as I told you, only the PS1 gonna involve, so there will be no formation of oxygen in case of the cyclic phosphorylation. Okay, so the 2 points you have to remember, in case of cyclic phosphorylation, because of the absence of dehydrogenase enzyme, only the ATP molecule will form, no NADPH. And the second point you have to remember, the photolysis of water gonna happen only in the PS2. In case of cyclic, only the PS1 gonna involve, so the photolysis will not happen, so there will be no evolution of oxygen from the particular photosynthetic system. Okay, so come back to the choices. Cyclic phosphorylation results in the formation of, okay, as I told you, ATP gonna produce, but there will be no NADPH and oxygen, so you can easily eliminate the choice B. As I told you, NADPH will not get produced over here. Okay, NADPH and ATP will produced only in the non-cyclic. Over here in case of cyclic, only the ATP molecule got released from the particular cyclic phosphorylation. This is how you have to understand the particular type of question and answer the question. Okay, now we will continue the questions from the same topic. And see the next question now. As compared to the C3 plants, how many additional ATP molecules are required to produce one hexose in C4? Okay, this is what your question. So this is talking about the production of one 6-carbon sugar. Over here, for example, glucose. Okay, so one 6 carbon sugar. Here they are comparing the C4 plants and C3 plants. Okay, and the question is about the number of ATP molecule. Okay, so as we all know, the major difference between the C3 and the C4, as I told you in the previous question. So over here in case of C4 plants, there are two stages involved in, one stage in the mesophyll cells and another stage in the bundle sheath cells. Okay, so in case of mesophyll cells, as I told you, the carbon dioxide is being fixed by means of phosphoenol pyruvate with the help of enzyme PEPCO and the formation of oxaloacetic acid, conversion of malic acid. Then it will be transported to the particular bundle sheath cell. Over here, from the particular malic acid, the carbon dioxide is being fixed to the or entered into the particular C3 cycle over here, that is the Calvin cycle. Then it will be converted back or come back to the particular phosphoenol pyruvate stage. Okay, so the point you have to remember over here, in case of C4 plants compared to the C3 plants, they required more energy for the particular process. This is what you have to remember. Okay, so as I told you, there are two set of reaction, one in the mesophyll cells and the another one is the bundle sheath cells. Okay, so in case of the process going to happen in the mesophyll cells, the fixation of carbon dioxide and the formation of oxaloacetic acid and the malic acid formation, over here they required 2 ATP molecules. Okay, 2 ATP molecules compared to the particular C3 plant process, because in case of C3 plants directly they will start with the ribulose bisphosphate and the enzyme rubisco. Okay, so over here they required 2 ATPs more for single carbon dioxide molecule. So the question is talking about 1 hexose, that means you have to fix 6 carbon dioxide molecule, then only you will get 6 carbon atoms. So over here, the answer over here, you are not supposed to choose 2, because as I told you, 2 ATPs required to fix 1 carbon dioxide. So you can easily eliminate this because we are going to fix 6 carbons. Okay, so 6 is not an answer. As I told you, you have to multiply this with 2 ATPs. Okay, so the answer is 6 into 2, 12, and you can easily eliminate the other choices. Okay, so that means 12 ATP molecules required more than the particular C3 plants. And see the next question, which pigment absorbs the red and far red light? Okay, this is your question. So whenever you are seeing the term, the absorption of light, red and far red light, okay, and these are all called pigments: phytochromes, carotenoids, chlorophyll and cytochromes. But here the question is talking about the absorption of light. That means that particular pigment should have the capacity to absorb the photons or light energy. Okay, and see the choices over here. The cytochromes are the pigments, they are going to involve. The cytochromes are the complexes, they are going to involve in the electron transport chain. Okay, so they have the affinity for the electrons. They are going to transport the electrons stepwise, so that they are going to create the proton gradient. They are involved in the production of ATP molecule. Okay, so cytochromes are nowhere related to the absorption of light. You can easily eliminate that. Carotenoids are also the pigments, but they don't have the ability to absorb the light compared to the other choice over here. Okay, chlorophylls are the pigments, but they cannot absorb the particular light compared to the phytochromes. Okay, as I told you in our previous section, phytochromes. Okay, so the chrome means colored pigment, chrome means colored, phytochrome means the structure which possesses the particular pigments, they can absorb the particular light. So the answer choice over here you have to choose over here is phytochromes. And see the next question over here, the correct sequence of cell organelle during the photorespiration. So as we have discussed in the previous section, the photorespiration, they are going to happen only in the C3 plants. Okay, so over here there are more than the particular organelle. As I told you, photorespiration normally going to happen in a chloroplast. We all know that chloroplast are involved in the particular photorespiration process. But to complete the particular photorespiration process, it needs support from some other organelles. The question is talking about the other organelles. Okay, the correct sequence of the organelles involved in the photorespiration, and see the choices: chloroplast, Golgi apparatus, mitochondria, chloroplast, rough endoplasmic reticulum, dictylosomes, and chloroplast, mitochondria, peroxisomes, chloroplast, vacuoles, and peroxisomes. Just remember one thing: the Golgi apparatus, they are not at all going to involved in the particular process photorespiration, so you can easily eliminate the choice 1. And the rough endoplasmic reticulum, we all know that they are not going to involve in the photorespiration, they are going to involve the synthesis of proteins. Okay, and so you can easily eliminate the choice B. And now you have to choose from the remaining choices: chloroplast, mitochondria, and peroxisomes, chloroplast, vacuoles, and peroxisomes. You have to remember one thing: mitochondria is essential to complete the photorespiration. Okay, so you can easily eliminate the peroxisomes and vacuoles. Peroxisomes also involved in the photorespiration. So the order, the sequence of events going to happen: it will start from the chloroplast and it will releases the carbon dioxide and it will enter into the mitochondria, and from there it will be processed and transported to the peroxisome and it will come out of the particular cell. So this is how you have to understand the particular question. The sequence of the photorespiration is chloroplast, mitochondria, and then peroxisomes. And see the final question now. The stroma contains. Okay, so what is stroma? Here we are talking about the photosynthesis, and this is talking about the stroma of chloroplast. Okay, so stroma is nothing but the liquid matrix, semi-liquid matrix present in the particular chloroplast. And this question is talking about what is the composition of the stroma. So as we all know, there are two types of major reaction going to happen over here: dark and light reaction. Okay, so over here in case of light reaction, they are going to absorb the sunlight energy as a photons, they are going to excite the molecules, they are going to release the electrons. By releasing the electrons, they are going to produce they are going to convert the particular energy into energy carrier molecules, ATP and NADPH or ATP molecules. Okay, and these ATP molecules and NADPH molecules, the energy carrier molecules, they are going to be utilized in a dark reaction. Over here in case of dark reaction, the carbon dioxide molecule is being fixed into carbohydrates. Okay, so this condition is called dark reaction. This is called light reaction because by using light they are going to produce ATP and NADPH molecules. But over here dark reaction, because there will be no direct involvement of light over here, but they depends on the particular products of the particular light reaction. Okay, and the point you have to remember related to the particular question: the light reaction always going to happen in the membranes of the particular chloroplast. Okay, so it will going to happen in a grana, thylakoid membranes and lamellar membrane in the particular structures of chloroplast. But the dark reaction going to happen in a non-membrane structure, that is in a semi-liquid matrix. That structure is called stroma. Okay, so the dark reaction going to happen in a stroma, light reaction going to happen in a membrane structure. Now come back to the question. Stroma contains, see the first choice, light dependent enzymes. There will be no requirement for the light dependent enzymes in the particular stroma, because as I told you, in the stroma only the dark reaction going to happen. So you can easily eliminate the choice 1. Ribosomes. Okay, ribosomes may be the correct answer because chloroplast also contains the ribosomes. The ribosomes may be present in the particular stroma, but this is not this is not going to related with our context we are going to discuss over here. So you can easily eliminate the ribosome over here. Chlorophylls, they are going to present only in the membranes, and the pigments they are going to produce involved in the photosynthesis. Answer over here is light independent enzymes. Okay, so there are so many enzymes, they are going to involved in the fixation of carbon dioxide into particular carbohydrate. Okay, and these enzymes are called light independent enzyme. As I told you, all the process going to happen in a dark condition, that is without the light, and they need the support from the light dependent molecule. Okay, so the answer over here is light independent enzymes. Okay, they are being filled in the particular structure called stroma. This is how you have to understand the structure of chloroplast and you have to answer the particular type of question. In the next class we are going to continue the questions from the same topic, photosynthesis. Dear students, and today we are going to continue the same section, the photosynthesis, and see the first question. Stomata of cam plants. Okay, so there are choices about their role and position in the particular plant system. Always open during the day and closes at night, open at night and closes at day, and never open. Okay, first you should know something about the cam plants. So what do you mean by cam over here? So cam refers to crassulacean acid metabolism. Okay, so it's a kind of photosynthesis system happening in some kind of plants. So normally these cam plants are present in a very dry conditions. Okay, for example, the cacti plants. Okay, so they belongs to the particular cam or crassulacean acid metabolism plants. So over here they have to adopt the particular dry condition. Okay, this is what you have to remember. Okay, so as we all know, what is the role of the stomata in the leaves over here? So what is stomata first? Stomata are the openings present on the plant surface, that is leaves. Okay, so there is a structure called aperture and it will be guarded by two guard cells. Okay, so they will open and close based on a plant system. Normally they are always open in during the daytime and always close during the night time. Okay, so why they are always open and during the daytime? Because they through the particular opening they have to absorb the particular carbon dioxide and they have to release the excess of water during the photosynthetic process. Okay, so this is what you have to remember about the stomata. Stomata is the opening in the leaves. Okay, and they'll always open during the day and they'll always close during the night. This is the normal thing you have to understand about the plant system. But as I told you, over here in case of the cam plants, okay, so I told you they are in a very dry condition. Okay, so they have to adapt to the particular dry environment over here. So they have to prevent the loss of water from the particular system. Okay, to prevent the loss of water, they have the adaptation. Okay, over here, based on that their stomata will function completely differs from the particular normal stomata. Okay, so over here see the choices. Never open is a completely wrong answer. They have to open at some time, then only they can do the particular carbon dioxide assimilation. Okay, so always open is also should be a wrong, because if they are gonna be always open, they'll lose excess of they lose excess amount of water and they cannot absorb the particular carbon dioxide. So both A and D are wrong choices. Now you have to choose between the B and C. Open during the day and closes at night. Open at night and closes during the day. Okay, as I told you, they have to adapt to the dry condition. They have to prevent the water loss during the sultry weather. So the answer choice over here is they are always open at night and closes at during the daytime. Okay, so this is how you have to understand the cam plants. So what kind of phosphorylation possible in the cam plants? As I told you, there are two types of phosphorylation. We have seen C3 and C4. Over here normally they will go for C4 kind of phosphorylation, C4 kind of carbon dioxide assimilation. Why? They have to get more, or they have to assimilate more amount of carbon dioxide to increases the biomass by using the available amount of light energy. Okay, and see the second question over here. Which of the following absorbs light energy? Okay, and there are choices for you: ribulose bisphosphate, oxygen, water, and chlorophyll. Okay, we'll see one by one. Ribulose bisphosphate is an acceptor molecule. We all know that. Okay, they are going to accept carbon dioxide. This is what you have to remember, ribulose bisphosphate is acceptor molecule, we all know. Okay, this is a 5 carbon structure, 5 carbon organic molecule. They are going to involved in a Calvin cycle, that is C3 cycle, and they are going to accept carbon dioxide, not the particular light energy. Okay, so light energy means over here from the sunlight the light will be traveled as in the form of photons, the packaged form of photons, and the photons will be absorbed by some form of structure. That is what we are going to see now. And the second choice over here is oxygen. Okay, so just remember one thing, oxygen over here in case of photosynthesis, oxygen is being released from the particular system. It is not going to be absorbed by the particular system. Normally the oxygen will be released or evolved from the photolysis of water. Okay, so the photolysis of water leads to the release of oxygen plus release of electrons plus protons. Both electrons and protons go and enter into the electron transport chain, and the further photosynthesis system and synthesis of ATP and NADPH molecule, and the oxygen will be evolved from the particular system. Okay, so both the oxygen and the ribulose bisphosphate are the wrong choices over here. Then you have choices chlorophyll and water. Okay, so you may get confused with the choice water. Okay, because as I told you, the very first step in the photosynthesis over here, the photolysis of water. So you may think that the water may absorb the light energy. That is not a correct choice, because water is being split over here by means of the energy utilized by the particular system. Okay, so what is the source of the particular energy? Energy is from the light source in the form of photons, and the photons energy form will be absorbed by some kind of photosystems, and the photosystem made up of the pigments called chlorophylls. Okay, the chlorophyll will absorb the particular light energy in the form of photons. They got excited, and the excited form will split the particular water molecule. After that they will release the particular oxygen molecule, and the electrons will be transferred to the further system, and the transfer of electrons will form the ATP and NADPH molecule, and then the formed ATP and NADPH molecule will be utilized for the synthesis of the carbohydrate. This is what the sequence of the process in the particular photosynthesis. What is the very first process? It starts from the light source, photons, and the absorption by the pigment system, chlorophyll, and the chlorophyll will lead to the excitation of the P700 or P680 system based on a photosystem 1 or photosystem 2, and from this only the electron transfer gonna happen, and before we are entering into the electron transfer, it will split the particular water molecule, and that will release the oxygen and other atoms over here. And see the third question over here, select the correct statement of the following. Okay, so one or two statement may be correct over here, and you have to read the statement and then you have to answer the particular type of question. Okay, and there are 4 statements given for you. Set scheme of light reaction takes place in presence of PS1 only. Okay, so to understand the first statement, you should know something about the set scheme. So what do you mean by set scheme? So as we have seen in our previous classes, there are 2 systems gonna involved in the photosynthesis, PS2 and PS1. Okay, so as I told you in a previous question, light, then photocenter, P680, and the excitation, the electron acceptor, okay, then electron transport chain, then again next sender, photocenter. In case of the PS1, the electron excitation, electron acceptor, then the formation of NADPH, and over here in case of electron transport chain, the formation of ATP. Okay, so this is what the sequence of event we have seen it now, and here you can see the shape, the Z shaped reaction. Okay, so that is why we are calling this as Z scheme. Okay, and the question, the statement over here, the Z scheme of light reaction takes place in presence of PS1 only. You can easily see it over here, involvement of both PS1 and PS2. So the first statement may not be correct, because as I told you, here the statement says PS1 only. This is wrong. Both PS1 and PS2 involves in the Z scheme. And see the second statement. Only PS1 is functional in case of cyclic phosphorylation. So we have seen in the previous classes, there are two types of phosphorylation possible in case of photosynthesis, cyclic and non-cyclic phosphorylation. So what do you mean by non-cyclic? So this system is non-cyclic. So over here the electrons will not be transferred back to the system. They will be finally transferred to the acceptor molecules and they will synthesize the particular ATP and NADPH molecule. Okay, so there will be no cyclic or rotation of the particular electrons over here in case of the particular non-cyclic phosphorylation. But in case of cyclic phosphorylation, only the PS1 system gonna involve. Over here the electrons will be cyclic and they will synthesize only the ATP molecule. Okay, this is what you have to remember. In case of cyclic phosphorylation, there will be no synthesis of the particular NADPH because the particular enzyme required for the synthesis of NADPH is absent over here. Okay, so that it will leads to synthesis of only the ATP molecule and the electrons will be transferred back or cycled back to the particular system. That is why we are calling this as cyclic phosphorylation. Okay, and see the statement, only the PS1 is functional in the cyclic phosphorylation. This statement is correct. And see the third statement over here, the cyclic phosphorylation results in ATP and NADPH2. As I told you over here, the enzyme NADP dehydrogenase is absent in the cyclic phosphorylation. So there will be no synthesis of NADPH molecule in the particular cyclic phosphorylation. Only the ATP molecules will be synthesized in the particular PS1 and cyclic phosphorylation. So the statement over here says cyclic phosphorylation results in ATP and NADPH. NADPH will not be synthesized over here, so you can easily eliminate the statement as wrong. And see the next statement, stroma lamella. Okay, stroma lamella, stomata lamella lacks PS2 as well as NADP. Okay, so this is what the statement given for you, and you have to remember one thing, the cyclic phosphorylation. Okay, so they are going to happen in a stroma lamella. That means they don't need the particular PS2 system because the cyclic phosphorylation going to happen in a particular area. What is stroma lamella? In case of the particular organelle, the chloroplast, the structure, the membranous protruding structure is called stroma lamella. So over here in case of the stroma lamella, they lack the structures called PS2 and NADP because as I told you, only the cyclic phosphorylation going to happen in the stroma lamella. They don't require the particular PS2 and NADP because they are not going to synthesize the NADPH molecule. They are not going to involve the non-cyclic phosphorylation. So this statement is correct. Okay. So what are the statements correct? Only the 2 and 4 is correct. Okay. So where is the 2 and 4? The first one is correct and the remaining are the wrong choices. Okay. This is why you have to understand the particular type of question. There are statements given for you and you have to read the statements very thoroughly and you have to understand the concept of the particular statement. Based on that, first you have to choose the correct statement, then you have to see the choices, and among the choices you have to choose the correct combination of correct statements, then only you have to answer the particular type of question. And see the next question over here. The electrons from excited chlorophyll molecule of PS2 are accepted first by. Okay, so this is what the clue for you. So over here you have to note down over here the system. They are talking about the photosystem 2 and they are talking about the electrons and they are talking about the accepting molecule, the first accepting molecule. Okay, so see the choices over here. Cytochrome B, cytochrome F, ferrodoxin and quinones. Okay, so as I told you, you have to carefully note down over here. This is talking about the PS2 system. So as I told you, there are some differences between the photosystem 1 and the photosystem 2. Okay, so in case of photosystem 2, over here the reaction center will contain the chlorophyll molecule P680. In case of PS1, they contain the particular system P700 chlorophyll system. Okay, so over here the sunlight energy absorbed in the form of photons. Okay, and then the molecular excitation will happen and the excited molecule will releases the electrons, and the electrons will be accepted by, okay, this is where the question arises over here, the accepting molecule from the particular photocenter. After the molecule excitation, the excited molecule will releases the electrons. So over here in case of photosystem 2, okay, so you have to remember one thing, the cytochromes are involved in the electron transport in the in between process. We have seen the set scheme, okay. So the electron transport happening in between the PS2 and PS1 over here, or in case of cyclic phosphorylation, it will happen within the particular PS1 system, but it is an intermediary process, not the very or initial stage of the particular photosynthetic reaction, okay. So the cytochromes are involved in the intermediary process, that is the electron transport chain. So you can easily eliminate the cytochromes B and F, and the remaining two choices over here, the ferrodoxin and quinone, okay. So in case of the photosystem 2, in case of PS2, you have to remember one thing, the first electron acceptor groups are the quinone molecules, okay, plastoquinone or phytoquinones, okay, they are gonna accept the electrons from the excited region. This is what you have to remember. Ferrodoxin is not a right answer, I will explain you why, okay. So quinone is the correct choice for the PS2 electron acceptor. And see the next question, this is a continuation of the first question. In PS1, the first electron acceptor over here, as I told you, you can easily eliminate the cytochromes and plastocyanin. They are the some other pigments involved in the photosynthesis, and again you have the choices among the two, ferrodoxin and iron sulfur proteins, okay. Just remember one thing, both the ferrodoxin and iron sulfur proteins are involved in the PS1 system. As I told you, the ferrodoxin is not going to involve in the PS2, okay. Ferrodoxin also going to involve in the PS1 only, okay. But the question over here is the first electron acceptor, okay. So the first electron acceptor in case of PS1 is iron sulfur proteins, okay. This is what you have to remember. Whenever you have a choice of the iron sulfur proteins, you have to choose this answer as a first electron acceptor. When you don't have the particular iron sulfur protein, then you can choose the ferrodoxin as a electron acceptor for the particular PS1 system. And again I will remind you, you are not supposed to choose the ferrodoxin as an electron acceptor for PS2 system, because ferrodoxin is only involved in the PS1 system, okay. So in case of the particular PS2 system, as I told you, from the excitation, the electrons will be accepted by the next system over here as, okay. So over here it will be accepted by the iron sulfur protein in case of the PS1, and the iron sulfur protein is a iron containing protein, okay. It's a iron containing protein. So over here it will accept the electrons, then it will transfer the electrons to the particular ferrodoxin. From the ferrodoxin it will be transferred to the downhill energy level, and that will produces the ATP and NADPH molecule over here, okay. So the point you have to remember, in case of the PS1, it will accept the electrons by means of the iron sulfur proteins. And see the next question over here, in case of the photosynthesis, energy from light reaction, okay, energy from light reaction to dark reaction is transferred in the form of, okay. So as we all know, there are two major reactions, light and dark reaction. In case of light reaction, the energy molecules will be produced, and the energy molecules will be transferred to the dark reaction. In the dark condition, they will fix the carbon dioxide to the carbohydrate molecule, okay. So the point over here, in which form the particular energy is being transferred, okay. So we all know that the ATP and the ADP molecule may be the correct answer, and you may get confused with the ribulose bisphosphate and chlorophyll, okay. Chlorophyll you can easily eliminate because it's a pigment, it is not gonna transfer the energy, it is gonna excite the molecule by absorbing the light energy, but they cannot transfer the energy, so you can easily eliminate the chlorophyll. This one is ribulose bisphosphate, this one is adenosine triphosphate, adenosine diphosphate. All the three components having the phosphate group, okay, they all can carry the energy, the electrons, okay, energy in the form of electrons. But the question over here, which form the energy is being transferred from the light to the dark reaction. So over here you can easily eliminate the ribulose bisphosphate because they are going to involve in the accepting of the carbon dioxide molecule. They are not going to involve in the transfer of energy from one step to another step. And you have the choices of ADP and ATP. ATP is being synthesized from ADP, but you cannot transfer the energy in the form of ADP. Okay, I will explain why. Adenosine, this is adenosine diphosphate, this is adenosine triphosphate. Okay, so this phosphate group is far from the particular adenosine. Okay, so you can easily break the particular phosphate group from the particular adenosine molecule. So the breakage of this molecule will release us the energy, and that energy will be utilized by the other reactions of the particular system. Okay, so that is why we are calling the ATP as a energy carrier molecule. You cannot store energy. Just remember this statement. You cannot store energy. You can carry the energy in the form of the ATP molecule. ADP molecule you cannot use as an energy carrier. Okay, as I told you, in case of ADP only the two phosphate groups are there. Okay, the energy required to break the particular phosphate group from the ADP will be very high. So you cannot use the particular ADP as a energy carrier molecule. So the point you have to remember over here, always you can transfer the energy in the form of ATP, not the ADP. Okay, so ADP required to synthesize the ATP. Okay, as I told you in the previous classes, ADP along with the inorganic phosphate, they are going to synthesize the ATP by using the proton gradient in the process electron transport chain, or the phosphorylation happens in the electron transport chain during the particular photosynthesis and in the respiration. We are going to see the respiration later in the classes. So the point you have to remember over here, for the particular type of question, in the photosynthesis, energy transfer from light to the dark condition over here in the form of ATP. Okay, this is what the answer. And see the last question over here. The first carbon dioxide acceptor enzyme in C4 plants. Okay, so this is talking about C4 plants. Okay, this is the first clue for you. And carbon dioxide acceptor molecule, this is the second choice, second statement or clue given for you. And over here you have to note down one thing. This is talking about enzyme. Okay, this is what you have to note down very carefully. Okay, this is talking about C4 plants, talking about carbon dioxide accepting molecule and carbon dioxide accepting enzyme. This is not talking about the molecule, acceptor molecule. This is talking about acceptor enzyme. Okay, so over here there are choices given for you. Ribulose diphosphate or ribulose bisphosphate is a carbon dioxide acceptor molecule. Okay, ribulose bisphosphate or ribulose diphosphate is a carbon dioxide acceptor molecule, organic molecule, 5 carbon molecule. They will accept the carbon dioxide and they will become the intermediate 6 carbon molecules, and they will produce 2-3 carbon molecules. Okay, so you may tend to choose the particular ribulose bisphosphate as an answer, but this is not a correct answer. As I told you, ribulose diphosphate is a molecule, but over here you have a clue, ribulose diphosphate carboxylase. Okay, but you have to remember one thing, ribulose bisphosphate gonna involved in a C3 plants. Okay, this is the acceptor in the C3 plants, and ribulose bisphosphate or diphosphate carboxylase and rubisco both are same enzyme. Okay, so rubisco is the ribulose bisphosphate carboxylase. Co refers to carboxylase and oxygenase. Okay, so both are same enzyme. So this is a kind of activity of the particular rubisco enzyme. Okay, so both the enzymes gonna involved in the C3 plants, not in a C4 plants. And remaining two choices, phosphoric acid and phosphoenol pyruvate carboxylase. As we have seen in introduction classes, in case of C4 plants, the primary acceptor of carbon dioxide is phosphoenol pyruvate, and that molecule, that reaction is being catalyzed by the enzyme phosphoenol pyruvate carboxylase oxygenase, that is PEPCO, phosphoenol pyruvate carboxylase oxygenase. Okay, so the answer over here for the particular type of question is PEPCO or the phosphoenol pyruvate carboxylase. Okay, So the complete name for the particular enzyme, PEPCO. So why we are calling this as PEPCO? Phosphoenol pyruvate carboxylase oxygenase. Okay, that is why we are calling this as PEPCO. Okay, so the carbon dioxide, first carbon dioxide acceptor enzyme for the particular C4 plant is PEPCO. Okay, this is what the answer. In the next class we are going to discuss something about the respiration in plants. Okay, then we can going to discuss the question we can expect from the particular topic.